So today we're going to look at the tubular function and all the things that happen inside a nephron. First, you want to know the basic sort of anatomy, where you have the glomerulus and the Bowman's capsule. You have the PCT, so proximal convoluted tubule, the thick and thin ascending limb, ascending and descending limb of the loop of Henle, and also looking at the distal convoluted tubule and collecting duct. So first we want to start off with these capillaries or arteries. So you have the afferent and efferent arterioles, and what happens here is we start off with some molecules going through the nephron, and here we can see that the concentration is around 300 milliosmoles and 300 milliosmoles here as well. So this means that this is isotonic. And what happens is anything that's going through the nephron is going to be moving through and turned into urine, and anything that's getting reabsorbed is probably important. So what I like to think of this is that if you have a lot of things in your fridge, you take everything out and then you put all of the important things in. So here you've just taken everything out, and then you're putting in all the things that you're like, oh, this is important, and the things that aren't important keep traveling down. So first of all, from the efferent arteriole, we get things like hydrogen ions and ammonium and creatinine, which get absorbed into the proximal convoluted tubule, noting that the proximal convoluted tubule is responsible for around 60 to 70% of reabsorption, and so it does most of the work. There's specific channels in here as well. So first you have the sodium and hydrogen channel, or the NHE3. And you also have glucose channels as well. So SGLT2, which is more in the early proximal convoluted tubule, and then the late proximal convoluted tubule has the SGLT1. So you have that, and noting that wherever sodium goes, water is going to follow. So these two are friends. And we're looking at the apical surface, so the basolateral surface, which is this one here. The basolateral surface, so if we draw this, basolateral means the ones that's close to the bloodstream, or whatever arteriole you're looking at, and then here you have the lumen of the kidney. So you'd have things like aquaporin channels in the basolateral side, and the sodium potassium ATPase pump as well. Now, when we're looking at the proximal convoluted tubule, this is permeable. It's permeable to water. So, and it's mostly paracellular as well. And in this part, angiotensin 2, we can just write that. This stimulates the reabsorption of sodium, so it can get reabsorbed into your arterioles. All right, moving forward, we look at the thick ascending limb of the loop of Henle. So there's a lot of stuff going on right here. And you have these channels, so the NKCC2, sodium, potassium, and this one is chlorine, and you have two of them, and it's a symporter or co-transporter. You also have ROMK channels as well, which move these potassium ions into the lumen, so it increases the lumen positivity and repels sodium, calcium and magnesium. So I haven't written magnesium here, but it also repels magnesium. So magnesium can kind of just leak out as well. And the ROM-K channel right here, we can write that, renal outer medullary potassium channel. This basically, or transporter, helps with that paracellular reabsorption as well. And this part of the nephron is impermeable to water because it dilutes tubular fluid and the medullary osmotic gradient is established. So this part is more hypertonic. Remembering that here we talked about how this was equal, so it's isotonic. Now we move upwards, and what happens is we move to the distal convoluted tubule, and you can see a lot of stuff happening here in those principal cells. So this accounts for around 5 to 10% of sodium reabsorption, and we use those NCC channels, or sodium chloride symporter or co-transporter. And these are very sensitive to diuretics like thiazide. So they target these, so thiazide diuretics, what they do is they target these channels, and this will help to increase calcium reabsorption. Now, an increase in calcium reabsorption can also be done by a sodium-calcium transporter. So we can see that kind of happening here. And this part is also impermeable to water because we don't have any aquaporins that are present. And its lumen is more hypoosmotic. So you can see hypertonic, hypoosmotic. And the parathyroid hormone can also act on these principal cells to increase, so it goes through the whole cascade of CAMP and protein kinase A and all that, to help bring in calcium, and this calcium can then be reabsorbed in those arterioles. To balance it out, we also have this hydrogen or proton calcium pump, and we get sodium out. Now, then we move on to the collecting duct. But before we do that, we can see that there is an effect of angiotensin. So first what we have is we have angiotensinogen. And this is basically a precursor. It uses renin to convert into angiotensin 1, and this converts into angiotensin 2 by using an angiotensin converting enzyme. And this happens in various parts of the body, but once you get angiotensin 2, it acts on the I think it's the medullary part, and it helps to secrete aldosterone. Now, after aldosterone has been secreted, it acts on these principal cells and changes like in gene formation. So it goes through a whole cascade, and it acts on your epithelial sodium channels and also aquaporin 2. So this causes sodium to be reabsorbed, and it causes potassium to leave and move through the nephron into the collecting duct. And you know, wherever sodium is moving, water is also going to follow. And this can help to increase your blood volume and your blood pressure. Now, we move on to the collecting duct. But before that, you want to understand some other types of principal cells, so intercalated alpha and intercalated beta. So this basically refers to acids. So alpha for acids, this refers to bases. So what do I mean by that? Well, you know how you have carbon dioxide reacting with water to get this, and this converts into carbonic acid, and this goes into bicarb, and bicarbonate can move into the arterioles, or the blood supply, and the hydrogen ions move out, and there's also a potassium that moves in. The reason why this moves in is so that there is still a balance and there's no change in the electrochemical gradient, but here we're too acidic and we want to get rid of these protons to become more basic. So if there is a significant decrease in pH, so acidosis, bicarb is brought into the bloodstream to bring the pH back to normal. Whereas intercalated beta cells, they are more basic. So you have too much bicarb, and we go through the same reaction, but in this time, we are taking out the protons and moving that into the bloodstream, and we're taking the bicarb and moving that into the collecting duct. We also have chlorine, which moves into these cells to regulate that electrochemical gradient. Because we've moved protons into the bloodstream, if there's a high pH, it will bring this back to normal. And another thing that happens is ADH can react on these receptors on the principal cells, which go through the whole cascade, and they can have effects on these specific vesicles. And these vesicles can act on the apical surface of the collecting duct, and that can create spaces for more aquaporin 2 channels. When there's more aquaporin 2, there's more water going inside these cells, more water going out. So these are, I'm pretty sure these are like aquaporin 3 or aquaporin 4 or whatever. But this water moving through the principal cells into these channels moves into the bloodstream. And this causes an increase in your blood volume and your blood pressure. Now, at the end of the collecting duct, everything that we didn't need gets excreted as urine, but there is some parts of urea that we still want to keep. The body is still like, we still need that. And so this urea moves down into this part here, just in the lumen. And this helps to excrete more water, essentially. We also have this blood supply here called the vasa recta. So if this is too positive due to the ROM-K channels, what happens is this some of these ions, they get excreted out into this bloodstream. Because it's too salty now, it gets rid of the water, because all of this salt, all of these ions are going inside. So these ions then move across, they move in, and then they get out. They get out and water goes in. And this is basically how your nephrons work. Another thing to point out is there's also things like drugs that also get extruded here, and then they can move through, and even things like ammonium, they can react with some water, so water and ammonium to become ammonia. And you could do it with water, you could do it with bicarb. We just need hydrogen to make ammonia, and then that can also then be excreted out into urine. But essentially that is how it works. You should also know that large proteins like albumin can't really go through into this whole thing, so that's why you have this sort of barrier. And this barrier in the glomerulus is really important because you have things like podocytes, and that's your parietal layer. And actually, maybe it's your visceral layer. So you have a parietal and a visceral layer in your glomerulus, and you want to know basically the process that it goes through, just to understand what can go through the glomerulus. So let's say that you have, I'm pretty sure podocytes are the visceral layer. So you have these podocytes here with foot processes and filtration slits. And so you go through that, you go through the glomerular basement membrane, which is most likely electronegative. And through that, you also have this simple squamous epithelium, which is your parietal layer. And that's mainly outside. It's probably like the thinnest layer, I think. But you need to go through all those layers just to get into the proximal convoluted tubule. So things like large proteins won't be able to do that. And there's different epithelium, as you can see here, around the proximal convoluted tubule. And this is simple cuboidal epithelium that you need to know. Also, the glomerular filtration rate of a normal healthy person is around 125 milliliters per minute, and this is around 180 liters per day. So you need to know those numbers.